Hi, and welcome to Ars Electronica Home Delivery. Right now we are here at the Ars Electronica Biolab, surrounded by all kinds of intriguing and fascinating scientific equipment, which is the perfect setting for another one of our artist and science talks. Today with our resident biohacker, Andreas Stürmer. Hi Andy, great having you with us.
Hey.
Cool. So you're going to tell us something about biohacking today. But before you start with your presentation, dear visitors at home, as always, you're invited to join our discussion here. Just post your questions into the YouTube chat and we will try to discuss and hopefully answer them. So without any further ado, Andy, the stage is yours.
Right. So let's start to pick up, to take apart the word biohacking, because hacking always sounds a bit scary to a lot of people, but in the main sense of hacking, it would mean taking something apart and making it, understanding the thing and make it do something new cool. Like the original biohacker or the original hacker said, I want to take my coffee machine and turn it into making a toaster. So when the media talks about do-it-yourself, when the media talks about biohacking, they always want to brand it as biohacking because it sounds dangerous, it sounds exciting, but in the end there's three different fields. There's do-it-yourself bio, which is more about education, community labs, do-it-together science. Then there's independent research, which is just doing research out of academia or the industry. And body hacking. Body hacking is, I mean, it pretty much says what it is. You hack your body, you modify it, and then there's overlaps, of course. If you imagine the Venn diagram, bioart can be both of body hacking and do-it-yourself bio. Startups can arise from do-it-yourself bio or independent research and so on. So I'd say we start with body hacking, which is typically this nowadays. So we're looking at an RFID implant, and there's lots of people nowadays who implant a magnet or a chip into the hand, and then they can open doors, for example, or their Tesla. Some more things, I mean, they get more sophisticated. You can make blinking lights. One biohacker took a Wi-Fi router into his leg, so he implanted it, or yeah, he didn't implant it, somebody else implanted it into his leg. I don't really know if that makes any sense, because, you know, it could, some people argue that the government can take it away from him if it's implanted, but I don't think that's really true, because they can cut off your leg and then you don't have your router anymore. Oh, and this is a very famous example, at least in the scene and in Australia. This is a guy called Miao Ludo Disco Gamma Mio Mio, and that's his real name. He was renamed into this. And he took the chip from the Opal card, which is the subway of Australia. And then he took the chip and implanted it into his arm or into his hand, so that he would be able to go into the train system and then pay with his hand. Obviously, he was convicted. There was controlling people. They controlled him, and he didn't have the card, and so he had to pay. And then he sued back. He was sued, and then he sued back. And I think I don't know if he won or not. But it's a case of cyborg rights, basically.
Yeah. So by adding one of those implants to your body, you're making yourself basically a cyborg, aren't you?
Yeah, I'd say so, because cyborg just means that you control or measure the environment with technical devices that you implant into your body that become part of you, right?
And do you know, is it legal to modify your body that way?
Honestly, I don't know. I think you can do it to yourself, but if I implant you, then okay. I'm not a doctor, and that's why you could sue me if something goes wrong or something. Okay. I mean, that's at least the case for a lot of experimental stuff, so that even an injection, if you inject insulin or if you inject whatever, or if you give you aspirin, then it would be...
Okay, but nobody can stop me from modifying my own body.
I don't know if the Krankenkasse, the health insurance, would say you made this yourself, I don't pay.
Okay.
But I mean, the same is like if you jump from a plane with the parachute and you hurt yourself, then it's also you did it yourself. So I don't know if self-harm would, if the insurance can drop out. I mean, they always do with houses, right? It's like, yeah, you're not insured for fires, but for meteors. Anyways, let's jump to independent research. The second field of biohacking. So this is Sebastian Kochoba. His passions are making beautiful flowers. He engineers roses to be blue and petunias. You can see here the leaves in a petri dish. We did some cloning here before. He's genetically engineering them to be blue with coral proteins. But he also, more than that, he's the first guy to have had a DNA sequencer in his home. So he was at home, posted a picture with his pyjama, and he was reading the DNA of a plant that nobody else has ever read before, the DNA.
So when you say independent research, what exactly do you mean? What makes a researcher independent?
I don't think there's really a definition of that, but I would define it as not being part of academia or the industry. So I mean, Sebastian, he pays mostly everything himself.
Okay.
But I mean, it's his passion, right? Some people have the hobby to have a Ferrari, some people like to ride horses. He likes to sequence the DNA of wild plants.
If I, for example, should discover this passion myself, what am I looking at? Millions, hundreds of thousands of dollars, thousands? So how affordable is do-it-yourself and independent research?
Yeah, it gets remarkably affordable. If you go on eBay, such devices like PCR device, everybody's heard PCR right now from some virus, right? So it's about $200, $400.
Okay.
Of course, then there's shipment from the US, which is going to be like $300, and import tax of the customs, which is going to be another $400, but you can still afford it. And if you live in America, you're in the wonderland. The nanopore to read the DNA is about $900 for the flow cell.
That's pretty cheap. And I guess we have to explore this famed PCI machine a little bit later.
Yeah. Oh, and here's another project of independent research. I think it's a really powerful project, and you can see it's done in a shed. So you can see the wood and the do-it-yourself stuff. And so what this guy did, it's on YouTube, by the way, he gave the mice a gene therapy so they would live longer and live healthier and make more muscles. He started out with a litter, so 10 mice, which were all from the same parents. They were all the same age. So they're very similar, and you can assume that they would live approximately the same amount of time. And he gave these 2 mice the gene therapy called follistatin, and follistatin is what I call the gym hormone. So when you go to the gym, you exercise, your muscle grows because of the hormone follistatin. What he did is he made a gene therapy of the follistatin gene. He injected the mice at the end of their lifespan, so when they were very old, and then he started seeing changes. They grew in muscle, they got more fur, they seemed to be reverse aging, they seemed to get more healthy, and the other ones died that didn't get the gene therapy. And so universities have done this before, of course. So they've done it in mice, in dogs, in monkeys. There's even studies in monkeys, and it just consistently works. They all live 30% longer.
Could you do the same with me, for example? Well, arguably, I already have enough fur, but I wouldn't mind having more muscle and living longer.
It's complicated. So technically, the science is already there. It's just unequally distributed.
Okay.
Arthur Clarke and stuff. So yes, there's a company called BioViva. They're selling the gene therapy for, I think, $500,000 or $2 million.
Wow.
So they're a real startup. he's doing something very, very similar. You could argue technically the same for all practical purposes. And I mean, creating the DNA, amplifying the DNA is something that's really cheap. So it's going to be like, maybe it would be $100 for a human dose, maybe 200. But of course, he's in America, right? So he just had to do a course how to ethically treat mice, how to anesthetize them so they don't feel pain. And that's all he needed. And in Austria, there would be, or in Europe, there would be 700 laws against this.
And can you detect this genetic treatment afterwards? So let's say I'm a professional athlete and I'm training for Olympia. I wouldn't mind growing some additional muscle mass. So.
Yeah, that's the funny thing. The Olympic dev company which does the Olympic Games, they outlawed CRISPR and genetic therapies.
Okay.
The problem is though, you modify a few cells. So like I modified 10,000 cells here and they're really tiny, you know how tiny a cell is. So if I modify these cells to produce a hormone that goes in my blood and distributes and makes me stronger, you would have to do a genetic test of exactly those cells. And if I put them here or there, nobody knows where to test and it's impossible to really test.
Wow, okay, that really opens up the proverbial can of worms, so to speak. Well, let's see what the future will bring us.
Yeah, I mean, I feel like this is really a cool project, doing real meaningful science, observing longevity. He also tested it on himself, by the way. So he's really confident in it and he's done animal studies. And so, yeah, like this is the goal for me, which I like. It's making humans live longer, live a healthier life, making them happier, right? And here's David Ishii again, another one of his projects, making CRISPRed dogs. So Dalmatians, these beautiful dogs here, they all have a genetic disease. So every Dalmatian, every purebred Dalmatian has a genetic mutation. And what that means is they all can't degrade urea. So the acid that comes from the bladder or is excreted in the bladder. And then the uric acid comes into the joints and then you get arthritis and other health diseases. And arguably in all other dogs, in all other mammals, the gene is the correct version, even in wolves, right? And so if you reverse the damage that the breeding has done, if you reverse the mutation, then you would have a healthy Dalmatian. And it's just one single base pair, one unit of the DNA that you would have to change. And he's working on making it accessible. So he wants to do it himself. He uses the sperm of dogs because he's a dog breeder. He knows how to do that. He adds the DNA to the sperm for the CRISPR. Then the sperm fertilizes an egg in a female dog, and then it suddenly should make a dog which has the gene repaired. So that's what he's working on.
So this is the second or third time you mentioned this CRISPR, and you said you can use it to repair a single gene in the dog's genetic code. How does it work? What is this CRISPR? What does it do?
Right, so it's you can see here this DNA coil here has lots of these subunits, these single base pairs as we call them. And a CRISPR is an enzyme from bacteria, like most of our tools that we use for genetic engineering. It's a tool from bacteria that can be programmed to cut at a very specific site. So, for example, here the code could be ATGCGGGTTA, so it's only 4 letters of the genetic code actually, and you program it to 20 bases very specifically, and then it's going to cut at that specific site.
Okay, and this allows you to repair a single gene in the hundreds of millions of base pairs in the whole genetic code of a dog.
And that's the cool thing why everybody is talking about CRISPR, because in former times, in the Stone Age, so like 7 years ago, there was, if you wanted to make a genetic mutation like this, if you know where the genetic mutation was and you had to genetically modify it to be the correct one again, if you wanted to repair a gene, it would have taken 10 to 15 years, hundreds of millions of dollars you would just randomly insert DNA and then you sequence all of the puppies and see which one has the mutations. And obviously this would take 10 to 15 years because you would have to do millions and millions of mice or dogs to make the mutation. And nowadays you can design this in 20 minutes maybe and do it. The longest time is going to take it how long the dog is going to be pregnant.
Okay.
So that's what takes the longest.
Wow. And you can do it at home in your proverbial garage with readily available equipment.
Yeah,
so
it's easy to use, but in Europe it's illegal, like everything with genetic technologies. Okay. Oh, but funny enough, if you wanted to do the same as he does, you could legally do it by putting the dog sperm into the sunlight or in Chernobyl, chemical mutagenesis, like radiation, and then, or you smoke near your dog and the DNA mutates randomly. You don't know where it mutates, but it mutates. That's how you get skin cancer from sunlight. And then you could make millions and millions of puppies. One of them is going to have the mutation, and then it's non-GMO, then it's not genetically modified in the law, but it's obviously like you modified lots of genes with the sunlight.
That's just ordinary breeding, I guess. You're trying to produce a lot of samples and then take the best ones and then replicate those to speak in scientific terms.
Yeah, okay. It's complicated. Like there was this European court decision. They wanted to decide whether CRISPR was GMO or not, whether we would be allowed to use CRISPR. And then they even said that radiation mutagenesis, like it's done all the time, actually is GMO, so they confirmed that it's GMO.
What is GMO?
Genetically modified.
Ah, okay.
But the problem is that all our food is genetically modified by radiation and chemical breeding and colchicine and stuff like that. And so they didn't outlaw it because it would have just, like, we would eat no food, we would eat grass all the time.
Okay.
So, yeah, the genetic laws are very, very outdated in that sense. This is another project that David's done. I mean, let's stop with David after this. It's not a presentation about him, but I really like this. This is a family tradition. Every Halloween, so the 31st of October, he's producing glowing pumpkins with his family. So you take the glowing genes from marine bacteria, because in the sea everything glows in the night. And then you take the genes, you put them into normal bacteria, you put them in the pumpkins, and suddenly the pumpkins start glowing. Also, don't do this in Europe, it's illegal. In America, it's beautiful. And there's a YouTube video on it, how to make it. Changing the topic to something completely different and maybe a bit gross for some people. Fecal transplants, right? This is Josiah Soiner. He had lots of enteric issues. He had issues with his gut, with his digestions. And what he did is he went to doctors and asked them, what can I do against this problem of mine? And the doctors, he's even a PhD, so he's a bioscientist, and he looked in the internet and the publications, in the literature, and he found fecal transplants that should work. But the doctor said, this is America, this is not allowed here, it hasn't been approved, there hasn't been clinical trials. We know that it's being used in Asia, but it's not legal here. And then he wasn't able to do it at a doctor, so what he did is he just did it in some random hotel room. So he took antibiotics to kill his bad bacteria, and then he ate the feces of a friend, basically packaged in pills.
So you're telling me that eating someone else's excrements is illegal in America?
Yeah, I would think also, even in Europe, like every kind of medical therapy has to be approved before you are allowed to give it. And I mean, this obviously counts as a therapy unless it's a sexual fetish, then maybe, I don't know where the borders are and how you could, maybe you can break the law, not break the law by circumventing it, saying, no, this is a sexual practice, not a medical practice.
Okay, and where did he get the antibiotics from?
Oh, you just in the internet, it's America.
Okay, so you go to the dark net and buy...
No, Amazon, I guess.
you can order drugs from Amazon that completely kill every
that's
every living germ in your body.
That's what we do in the agriculture. We just kill all the bacteria and breed resistant germs with breeding animals, chicken, pigs, stuff like that. It's crazy that we can do this, but
wow. Okay. And do you have to care for compatibility? Does he have? Did he have to look out for a?
No, you just take tetracycline, which is a broadband antibiotic. It just kills everything.
Yeah, and as far as the excrements are concerned, so do you need someone who is particularly healthy? Or?
Yeah. You should. In countries which do that, you have to screen the donor. You screen the guy who, gives away the thing.
Is there a difference between male and female excrements?
I don't know. I just know that some people are apparently, I mean, it's a broad field which is being explored right now, so we don't have truth right now. But some people seem to be very, very happy from certain bacteria. So the bacteria produce hormones. That's why we call the gut is the second brain.
Okay.
The gut feeling, right? And so the bacteria produce hormones that make your brain work better or happier.
Okay, so in theory, I could genetically modify myself to grow more muscles, and I could eat someone else's excrements to be more happy.
Yeah.
Okay.
Well, unless you already are very happy and you have the good bacteria already.
Okay, and then additionally I add some implants to make myself a happy, muscular cyborg, so to speak.
Yeah, maybe.
Okay, sounds like a lot of fun.
And here we've got the third area, which is do-it-yourself bio. So maybe remembering we had independent research, we had body hacking, and now we've got do-it-yourself bio. And this is more the educational play around in community biolabs. Foundry, for example, is a very famous one in Australia. They've done a corona test with LAMP, which is superior to PCR. It's not yet approved or anything, but it's a very, very nice idea. And people are working on it. There's the Open Insulin project. Maybe you've heard, if you're in Europe, you're lucky. Insulin is not that expensive. The insurance covers your insulin. If you're in America, sometimes they charge $300 for such a tiny vessel or vial. And people sometimes die because they don't get insulin in America because they ration it. They don't take everything. They want to take less and less, and suddenly, you can die from that. They want to use phages this is really a cool sammelsurium of projects. Agrophages, which means you take bacteriophages to kill bacteria that are resistant to all our antibiotics. This is something that is certainly very interesting. Like viruses attack humans, there are specific viruses that only attack bacteria, and they have been harnessed. They have been used to kill antibiotic-resistant bacteria before. Lab-grown meat, that's all these kind of things that they do. Yeah, and this is maybe the most recent stuff, LAMP, as opposed to PCR, because PCR is an old technology, it works really great, but the LAMP is something similar. But it's easier to use. You don't have to use expensive machines like the thermocycler that we have here. And the result is it glows or it doesn't glow. If it glows, then corona is present. If it doesn't glow, then there's no corona present. So that's pretty cool there. And that's the PCR result. So you have to use a gel. You have to use all these devices. Here we've got the PCR machine.
And what is this? Man, it looks interesting and all, but what are we looking at here?
So this is a tube, and you put in the polymerase, the DNA, or the sample. So I can spit in there, for example, and then you add the reagents, and then it amplifies pieces of the DNA or RNA of the virus. And then you can see, okay, there is corona present or there's no corona present.
Okay, so I read somewhere on the internet that the very first corona PCR tests, they suffered from bad primers. So
yeah, it was made by the intern.
What is a primer and how does this work? So
let's see how we do this. Maybe I'm gonna pull up real quick a photograph.
Okay.
Is this a touch screen?
Yep.
Okay. No signal. I think it died.
Okay, you killed.
Oh yes,
you
yes.
killed on that note.
So, and the thing is not working. Okay.
Okay.
Primer. Unexpected question, but it's important to know what primers are. So,
just take care you don't delete the internet.
oops. Okay, so if you Google for primer, that always happens in biology. You find whatever this is, skincare cream, primer, PCR. Because 99% of the people look for primer skincare and not for DNA stuff. Here I got something that I like, I think. Let's open it. Google used to be used to open the pieces.
Asking those inconvenient questions.
No, inconvenient questions are great. Anyways, here's your template DNA. You can see the primers here and here.
Yeah, touch screen.
Yeah, touch screen. Okay. Yeah, these things here, they are like bookmarks, and this is where it attaches. They're short synthetic pieces of DNA. You can buy them for $7 online. And then they bind, and then the machine or the cell amplifies the DNA. And
by amplifying, you mean replicating?
Yeah, you make more of it.
Okay.
Right. And this technology has totally been stolen from bacteria, from human cells.
Okay.
We just took it and put it in the machine, and it does the thing.
And what is different with this newfangled lamp you were talking about?
So the lamp also uses some kind of priming devices. It folds loops, and then at the same temperature, it keeps amplifying the thing, as opposed to the PCR, which needs thermocycling, so it needs to go between temperatures.
Okay. So let's have a look at our live stream if there is any questions posted on the YouTube chat. Just give me a second. No, doesn't seem that way. No questions. Let's see if we have any more questions prepared.
Cool.
Yeah, the Biofoundry. Let's see if we can get your presentation back.
Yeah.
Like
So
here.
Here we got it.
What is Biofoundry and how would it help me if I were interested in doing yourself biology?
Yeah, so basically there's always a hurdle, right? If you're thinking about fab labs, 3D printers, laser cutters, they all cost money. And so there's people, university students, who said, let's come together and build a lab together. So I don't have to pay $50,000 for my lab, or 20,000, depends on if you're good on eBay or not. But you don't have to pay $20,000 for your lab. You can have 70 people or 50 people, and they all pay together, and everybody just pays a little.
So it works like a subscription, so to speak? I
Yeah.
pay a monthly fee and then I can go there and use all these fancy scientific machines.
Yeah, it depends on the lab though. Some of them, every one of them has like access, like they define who can come, and then, I mean, most of them are students anyway at the university, but if you're not student at the university, they want to know what you're doing and if it's safe and stuff. I mean, most
Do
of
we
the time
have
it's
something comparable here in Linz?
No, we have something in Graz,
Okay.
but in Europe, generally speaking, in Europe, it's just very, very too much paperwork. So like you have to certify your lab room, your lab space, you have to get approval for each individual experiment. And so sometimes it's experiments like making the bacteria glow, right? what my friend is doing there with his 14-year-old daughter, and people do it at American high school every day. And here in Europe, we need special certification and approval and stuff like that. So it's, if you're in the field, it sounds ridiculous.
So the Ars Electronica Biolab,
Yeah.
this experience going on here, this bubbling stuff and over there. So are we certified to do this?
Yeah, we do have an S1 certification, and it's also, we have the possibility, or we let artists in, bio artists and other people, so that they can come here and do work here.
Okay.
Again, like, you don't want it to be too elitist that only big corporations, big universities do the research, but also like if somebody says, I'm an artist, I want to learn about this stuff, I want to try out something new, he can come here and do stuff. And I think that's really important, so that not only big companies like pharma companies, I mean, obviously they can just pay people to do the research, they can pay people to do the job, to get certifications, to do all the paperwork. But yeah, sometimes it's small people asking the stupid questions that lead to research.
Yeah, and I think that's a perfect closing statement for today's science talk. Thank you very much, Andy, for having me today, and thank you, visitors at home. Tune in next time. See you, and bye-bye.